Hey, what's up, class? I'm recording this from Ann Arbor, Michigan. You guys might remember me saying that I was gonna head out to Ann Arbor and visit my family, my wife's family. She has a sister and a brother, and their families they live up here. Um, Her brother-in-law is a pastor, and he planted a church up here in Ann Arbor. Super cool. Um, but that's one of the actual awesome things about having a hybrid class. I will admit, as much as I hate not seeing you guys at least twice a week, um, the nice thing is that I can go out of town and still record the lecture and post it, and it's all good. So we're gonna finish, not chapter six. We're gonna finish our lecture from Tuesday, and actually, that's it. I'm gonna assign homework on that portion of the lecture that is gonna be due on Tuesday, and then we're gonna continue with the rest of chapter 6.6 on Tuesday. So let's get started. Here's a little overview of chapter 6. So first, we've already looked at what is cancer, that it's really not a disease from the outside, but it's really just your cells dividing uncontrollably because they've acquired mutations in important genes. And then we looked at why cells divide, the importance of cell division, right? You should know that you go from all of us started as one cell in our mother's womb, and that cell divided millions of times, and it's still and there's still a lot of our cells are still dividing. So cell division is important for growth. It's important for repairing wounds, for replacing old tissue. It's important for asexual reproduction in asexual organisms. They just divide, and you have another organism. We also looked at what happens to DNA when cells divide. So, remember, DNA is the blueprint of the cell, so it's really important to keep it intact and safe. So, how do cells do that during cell division? Especially when it is a spaghetti, a silly string of DNA within the cell. So, we looked at all that. We looked at DNA replication before a cell divides. The cell has to replicate its DNA so that each daughter cell has a copy of DNA. And then we looked at DNA condensation, that you have to take that silly string and condense it into chromosomes that you can separate neatly. And on Tuesday, we started looking at the cell cycle and mitosis. We looked at how cells divide, the 4 steps of mitosis. We'll kind of go over a little bit of that on this lecture, and we'll also look at how the cell controls cell division. And then next week, we're going to finish chapter 6 with meiosis, which is a special type of cell division that happens in our sex cells. And specifically, we'll answer the question, how do we pass on our genes to the next generation? All right, so let's finish up 6.3, finish up mitosis, and then we'll look at how the cell controls the cell cycle. Okay, so just a little brief overview. You have 4 steps in mitosis: prophase, metaphase, anaphase, and telophase. I encourage you guys to come up with a little acronym to remember, just so that you don't have to worry about recalling that information for the test. And you start with the interphase, which is what's happening before mitosis, right? So if you look at that little cell cycle diagram, the interphase is the G1, the S, and the G2 steps. So right at the end of interphase, you have replicated the DNA. So there you see in the nucleus, and the nucleus is in purple and the DNA is in red. You have replicated DNA that is not condensed. And you also have centrioles that have been replicated. So you have 2 of them now that will be important for making fibers called microtubules, which will help separate the DNA. So the first step of mitosis is prophase, this step here. And this is where I gave you guys keywords in class, so I hope that you remember them, write them down. They're also in the slide before this one on the lecture, the slides online. So basically in prophase, DNA condenses and the nuclear membrane or the nuclear envelope breaks down. In between prophase and metaphase, Another thing that happens is that these spindle fibers called microtubules attach to each chromosome at the centromere. And the centromere is simply the center of the cell. And in metaphase, the second step, what you see is that the chromosomes align at the center. And so they have done a little dance and now they're nice and neatly aligned. And in anaphase, you separate the replicated chromosomes, because remember, we'll talk about this more on Tuesday, but the chromosomes were replicated, and now what you're doing is you're separating each of the replicated chromosomes so that one end of this cell, that one end of the dividing cell is going to become its own cell and have its own copy of DNA, and so will the other end of the cell. In telophase, you basically undo what you did at the beginning. So you decondense DNA and you reform the nuclear envelope. So you should see the chromosomes already starting to look like silly string, but they don't show that here. And then you're done with mitosis. Remember, mitosis has only 4 steps: prophase, metaphase, anaphase, telophase. You're done with mitosis, but you still have not divided the cell. So now you have to go through cytokinesis. And cytokinesis, cyto means cell, kinesis means movement. So this is where you actually separate the two cells. And now you have 2 cells that can begin the whole process over again. So there, each cell is entering into interphase. All right, so let's put what we learned about DNA replication and condensation into context. So this is a slide that we already looked at. This was from the online lecture last week. And what happens is DNA replicates while it's still not condensed. So this is uncondensed DNA. This is chromatin, all right? You can think of it as an unwound chromosome. It's going to become a chromosome. And it replicates, and now you have 2 identical chromosomes. And those are called sister chromatids. And they interact at the center, called the centromere. And so this all happens in interphase. This happens before cell division. And now you enter into cell division. So you take that replicated DNA, and the first step in prophase, you condense DNA. So that DNA condenses, and now you have a legitimate chromosome. And this is a replicated chromosome that consists of 2 identical sister chromatids. And that replicated chromosome is going to go through the rest of mitosis, go through metaphase, align at the center. It's going to go through anaphase, separate, and then it's going to go through telophase where the nuclear envelope reforms. And you're going to have now identical chromosomes, 1 in each cell. I really encourage you guys to watch this video. This is a crash course video. Highly recommend Crash Course, Khan Academy, other YouTube resources that animate a lot of what we're learning. So follow the link. This video of Crash Course will take you through the steps in mitosis, animate it for you so that it can come to life, and that's going to help you remember this a lot better. Okay, so we wanted to mainly look at mitosis in this last set of slides. Now we're going to look at cytokinesis. And so cytokinesis is the division of the cytoplasm, or the division of the cell itself. The way I would describe it is mitosis is the division of the nucleus, cytokinesis is the division of the cytoplasm. And this occurs after the last step of mitosis, after telophase, and the cells split into 2 cells, and then each cell re-enters the interphase. In animals, what happens is that there are proteins that start forming between the 2 cells, and they begin pinching the center, almost as if you had a string that you're tightening, a little loop around the 2 cells there, and you're just going to tighten that loop until it pinches off. So there are proteins that do that tightening and then eventually pinch the cells off into 2 new cells. In plant cells, it's a little different. In plant cells, remember, they have a cell wall. So a cell wall is rigid and tough. Remember, cell wall is made out of cellulose, so that is why wood is so hard, because cellulose is a tough material. So then how does a cell divide? It'd be like dividing two brick walls, right? Really hard. So what happens is that within one cell, you have mitosis where the nucleus divides. And after the nucleus divides, you begin forming a plate, a new cell wall between the two new daughter cells, between the two nuclei. So in this figure here, you have two nuclei that have already divided within one cell. And within that cell, you begin forming that cell wall, which is made out of cellulose. And then eventually that cell wall is built and separates the two cells. All right, so a couple of questions from you guys, for you guys. First question: in some fungi and slime molds, mitosis may occur without cytokinesis. What would you expect to find in these species? So in these organisms, the fungi and the slime mold, mitosis is occurring, nuclear division is occurring, but cytokinesis or cytoplasmic division is not occurring. So what would you expect to find? Cells that don't contain nuclei, cells that contain multiple nuclei, DNA that never condenses into visible chromosomes, or nuclei that never enter interphase. Okay, gave you a little time to think about it. Let's go through some of these real quick. So A, cells that don't contain nuclei, that doesn't make sense because mitosis, if mitosis is occurring, that means that there's nuclei that are being divided, right? Mitosis is nuclear division. So A kind of doesn't really make sense. B, we'll talk about in a second. C, DNA that never condenses into visible chromosomes. Again, if mitosis is occurring normally, then the DNA is condensing. If the DNA condenses during prophase, so you know that C is not right because the cell does go through prophase. It goes through mitosis. What about D? Nuclei that never enter interphase. Could be. Very possible. But we haven't really discussed that. So that would be an iffy one. So if this is a multiple choice test and we're answering these questions, I would have crossed out A and C and put a question mark around D. And then B, cells that contain multiple nuclei, that you can know. Because imagine it, say one cell then goes through mitosis, the nuclei divide, so you have two nuclei within that one cell, but if it never does cytokinesis, it never separates the two cells, right? Cytokinesis is a separation of the cytoplasm, the division of the cytoplasm into two cells. So if you never do that, you end up with a cell that has two nuclei. And if you do that again, you end up with a cell with four nuclei, and so on and so forth. And yeah, these slime molds and fungi do this. So they have multinucleated cells. And our muscle cells actually do this as well. All right, interesting, right? How about this one? During which phase of the cell cycle is the DNA in the chromosome duplicated? During which phase of the cell cycle, basically during which phase is the DNA replicated? And by now, you should be comfortable and you should be able to say interphase. B is the right answer. For those of you that have been reading and chugging along, what step of interphase is DNA replicated? G1, S, or G2? Hopefully you know that S stands for synthesis, which is synthesis means to make something, and what you're making is DNA. So DNA is replicated during the S phase of the interphase. All right, now we'll look at a few, wrap up this short online lecture with understanding how cells control cell division. So there's this cell cycle, right? And I told you guys we've looked at how cells are really like little machines. They really are. They are so complex, so complicated, and they function like clockwork. Mitosis is an example. It's clockwork, right? And so the cell has to be able to control all of this, and there's a rhyme and reason to all of it. It doesn't just happen randomly. The cells actually control when cell division occurs, and can actually stop cell division if something is wrong. So the cell cycle has security checkpoints at each point to ensure that everything is okay, much like a TSA security guard at a security checkpoint at a airport, checking that everything is all right, that the people that are entering the airport are all clear to proceed to the next level. So that's essentially what these checkpoints are for. And so you have proteins, which are like the TSA security guards, that are looking at things like, is the DNA okay? Are the chromosomes attached to microtubules at metaphase so that you can begin separating them? Is cell in the beginning, before the cell begins to prepare for mitosis, the TSA security guard proteins are asking, is cell division even necessary? Are there signals telling us to divide? Is the cell large enough to enter G2 or to enter the S phase? Basically, in G1, the cell grows and prepares for the next steps. So at the end of G1, it asks, has the cell grown large enough? So yeah, so you have that checkpoint at G1 asking, is it ready to continue with preparation for division? At the end of G2, right before you enter mitosis, there are proteins that check the DNA and make sure that it was replicated correctly. Because remember, the DNA is a blueprint of the cell, very important. And then during mitosis, you have another checkpoint at metaphase, where the cell makes sure that all of the chromosomes are attached to microtubules, to these fibers, and they're aligned at the center. And it won't continue with cell division, with mitosis, until all of the chromosomes are at the center. And what cancers do is that they disrupt these cell cycle checkpoints. They're basically mutations that are affecting those TSA security guards. And so those security guards, it's like they're blind to people carrying guns into the airport, right? Something like that. Like, these security guards now can't tell whether the DNA has been replicated correctly. It might not be, and that's not good. A cell shouldn't continue with cell division if the DNA was incorrectly replicated, because you're introducing mutations, right? So the mutations in cancer can affect two main types of genes. So I'm going to compare these genes to brakes and gas of a car. All right. So the brakes and the gas are what allow the car to move forward, which basically is the cell going through the cell cycle. So mutations affect tumor suppressors, which are genes that suppress growth and division. So these genes stop the cell cycle whenever things are funky. They're the TSA security guards that we're talking about at each checkpoint. And they're the brakes of the system. So when anything is wrong, whenever you have a suspicious passenger going through the security checkpoint, it's going to stop things and make sure everything's okay. So what mutations do is that they inactivate the tumor suppressor, and the tumor suppressor can no longer suppress uncontrolled growth and division. So here you can see the normal protein. There's a tumor suppressor gene, which is at the DNA level, which encodes a tumor suppressor protein. And this protein is the TSA security guard. Now, if the DNA acquires a mutation, so you can see there's a mutation now in the gene, in the DNA sequence, that's going to affect the protein sequence, and it's going to most likely, mutations are almost always detrimental, so it'll most likely negatively affect the protein's function. So the tumor suppressor can no longer stop the cell from continuing through the cell cycle, and that cell can eventually become a cancer cell. Not from the beginning. It takes a little more than it's like cutting the brakes from a car. That's not going to necessarily make the car go fast, but it's going to prevent the car from stopping if it needs to stop. And the second class of genes are proto-oncogenes. Proto simply means before, and oncogenes are genes that have to do with cancer. Onco is a root word for cancer, so oncologist, is a doctor that studies cancer. And these are genes that promote growth and division. These are good genes. So they're the gas of the system. They're the gas of the cell cycle. Whenever the cell needs to go through the cell cycle, it steps on the gas, it activates these proto-oncogenes, which are good, right? So they help the cell go through the cell cycle. But what mutations do is they hyperactivate these proto-oncogenes, and then these become oncogenes. So these are now the cancer genes. So proto-oncogene just means before they become oncogenes, and once they acquire a mutation that hyperactivates them, they become oncogenes. So now you basically, if you look at the figure, there has been a mutation at the DNA level which has changed the protein shape. And now this mutation is constantly on. It's hyperactive. So it's like you're pressing the gas on your car always. And the terrible thing is, if you have that going on and you can't press the brakes, you can die. You can crash. And I guess in this analogy, that cell can become a cancer cell, can become a tumor eventually. So this alludes to the fact that cancers require multiple mutations or hit. One isn't enough. It requires at least 2 hits. So you have to hyperactivate proto-oncogenes, which turns them into oncogenes, and it's like pressing the gas. So you're starting to go fast. But you still have the brakes. The brakes still work. So as if the cell realizes that it's going too fast, it's going to hit the brakes and stop the cell division. And if it can't fix what's wrong, that cell's going to actually die. It's going to kill itself. There is a program in the cell called apoptosis, where the cell, if it's not doing what it's supposed to do, it'll kill itself. So if you look at the figure real quick, you'll notice, okay, on the left you have a normal cell where you have the nucleus, you have DNA, there's a normal tumor suppressor protein which repairs damaged DNA and stops the cell cycle if it needs to stop. And you also have a little protein receptor, which is the green little arm that's reaching out from the cell, that is binding to a growth factor. So this protein receptor would be an example of a proto-oncogene, because what happens is that your brain and other cells can release molecules called growth factors that tell cells to begin dividing. And so this growth factor will bind to this protein receptor, and the protein receptor will relay the message to the cell telling it to divide. Now, if this protein receptor acquires a mutation that hyperactivates it and turns it into an oncogene, basically what's happening is this protein receptor is basically on mode all the time. So it's telling the cell to divide all the time. And typically that won't even be enough to form a tumor, but in this example here, this is from a previous edition of your textbook, the cell forms a tumor. So you're pressing the gas, basically, right? And now it needs another hit to become a full-blown cancer. You need to inactivate the tumor suppressor or kill the brakes, because again, you are pressing the gas after hyperactivating proto-oncogenes, but you can still press the brakes and stop the whole thing. So what happens in these cells, when a cell acquires another mutation that kills the brakes, these are the cells that are going to become cancerous cells and that are going to form tumors, and eventually those tumors, as those cells divide, they acquire more mutations and eventually become invasive cancers that can invade tissue and spread throughout the body. So this is like killing one bird with 2 or more stones, right? It's the opposite. The saying is killing 2 birds with 1 stone. Here you need at least 2, if not more, stones in order to kill this one bird, this cell. Or not kill this cell, but in order to turn this cell into a cancer cell. So that's it. This is where we're going to be stopping. I'm going to be assigning homework on this, so make sure that you read through the textbook, understand this stuff. Let me know if you have any questions, and then go through the homework assignment. And I'll be seeing you guys bright and early Tuesday morning. I'll be handing out, some of you weren't able to stick around and wait for me to pass the test around. I apologize just for my unpreparedness this week, but we should be back on track next week. But all right, I hope you guys have a great weekend, and I'll see you on Tuesday.